ganglion cells. So in this particular part, we are going to look at types of ganglion cells. So this is what we are looking at, the types of ganglion cells. So I want to inform you that according to the textbooks I've read, I've found out that they talk about different types. There are about 6. I don't know how these guys identify them and group them, but this is how we are going to handle them. So we have what we call P cells. This is a type of ganglion cells. We have what we call M cells. We have K cells. And in this same bracket, we have what we call melanopsin. So this is one set of grouping. And then another grouping, we have what we call W, X, and Y ganglion cells. This is another grouping. So if we are to start with this grouping, I will find that P cells, these ones are the easiest to remember. This is that I look at M, and remember M goes with M, which is for motion. So we'll start with M cells since they are easier. So M cells, these are ganglion cells, are mainly responsible for detecting motion, and they mainly receive information from rod cells, not exclusively, but mainly from the rods, and their information is mainly for motion. Another thing is that you need to take note that they are the least among the types, the least number of cells. Then the other one, they transmit signals at a very fast or rapid rate, fast conduction. Their conduction is fast compared to these others. So mainly for motion, from rod cells, they are the least fast conduction. Maybe to add on that they carry no information concerned with color. That is, they're just responsible for white and black vision. What we would refer to as being achromatic. Achromatic meaning they don't carry color. So I'll write it here that they are achromatic. Chromatic. Then we look at P cells. These ones receive information mainly from the cones. And of course that would mean that each would be responsible for color vision. So they carry information concerning color vision. However, their conduction is slow. Conduction is slow. The other thing is that we know that cones are concerned with color vision and fine details. You get color vision and fine details. So, but how does this fine detail come about? It's as a result of 1-to-1 ratio of their convergence. You find that one ganglion cell synapses with one cone cell, you get? Which would imply that P cells will be the majority since we are having one-to-one, unlike the M cells which we see from rods where we have a synaptic convergence gate with the retinal convergence. So these are the most numerous, contributing more than 80% of the total number of ganglion cells, more than 80% of the total. You get? And since we have color, so these are chromatic ganglion cells. They transmit their signals at a slow rate, very numerous, and also concerned with the fine detail. Then the K cells, these one receive information about mainly what we call the blue-yellow color. Blue-yellow color. So they are mainly concerned with the vision of the blue-yellow color. Right? Those are the K cells, and they contribute about like 10% of the total. Then melanopsin is another type of ganglion cells. These are just concerned with the circadian rhythms. Circadian or I don't know how to pronounce it well, but circadian rhythms, those are the cycles, like the sleep-wake cycles. That is the melanopsin. So that's one grouping. The other grouping is the WXY. So according to this grouping, they tell you that the W ganglion cells, these ones will be responsible for transmitting information concerned with directional movement. You get? Directional movement or motion. Directional movements. Then X cells, of course, directional movements, you can also tell that it's about roads, as we have seen before here. About roads. Then X cells, these are for color, vision, and fine detail. That is for color, vision, and fine details. And finally, we have the Y cells. So these ones will be responsible for rapid changes in vision. You get? Rapid changes in vision. Rapid changes in vision. So we have directional movements for W cells, we have color vision for X cells, and rapid changes in vision for the Y cells. So those are the types of ganglion cells, and with that you have the entire knowledge of ganglion cells. And I want to summarize with bipolar cells. So bipolar cells, they are of two types. We have those that are depolarizing, which means upon stimulation they will depolarize. Then there are those which upon stimulation they will hyperpolarize. So these that depolarize are the ones that will be involved in the transmission, whereas hyperpolarization will inhibit transmission. Not forgetting that we have these two other cells, which are the horizontal and the amacrine cells. These play a big role in the enhancement of contrast. So I believe, having made it to medical school, you must have covered something about horizontal and amacrine cells. So these are the cells that will provide the lateral inhibition. So they release neurotransmitters like the GABA, you get, which will inhibit nearby cells so as to have a contrast in one particular bipolar or ganglion cell, respectively, where they are found. Horizontal for bipolar, then the amacrine for the side of ganglion cells. So with that, we've had a clear knowledge about the ganglion cells, And then we are going to look at the next part. We are going to look at the lateral ganglion nucleus and the visual cortex. We look at their layers, and we are going to find that their layers are namely the P layer, the M layer, the K layer. So the P layer will receive information from the P ganglion cells, the M layer from the M cells, and the K layer from the M cells.